Welcome everyone, or welcome back to the GHGA webinar. Today we're going to be covering good statistical practice for the analysis of large biological datasets with Wolfgang. We always like to advertise our next seminars, so if you're interested in either a beginner's guide to single-cell RNA-seq analysis or the fair management of biological research data, these are our next webinars coming up. We highly invite you to sign up for these, either using the QR codes or going to our website, ghga.de/events, and sign up for those. In case you haven't been to one of these webinars, I want to briefly introduce to you what GHGA is. It stands for the German Human Genome-Phenome Archive, and our mission is to provide infrastructure for GDPR-compliant human omics data sharing, to allow for secondary use of clinical omics data in research, which could enable further biological discovery and replication of findings, and further translation of research insights into genomic medicine. And since this involves a lot of interaction between very diverse communities, be it computational biologists, geneticists, clinicians, physicians, and patients, we aim to get a lot of people of different topics together in these webinars. And with that, I would like to welcome and introduce our speaker today, Wolfgang. He received his PhD in statistical physics in 1998 from the University of Freiburg, and did his postdoctoral research at the IBM in San Jose and at Datasets in Heidelberg. Has been group leader at EMBL EBI from 2004 to 2009, and then moved over to EMBL Heidelberg, where he is now group leader of the Quantitative Biology and Statistics group. He is the co-director in the Molecular Medicine Partnership Unit and also co-spokesperson for GHGA. And in 2018, he first published his book, Modern Statistics for Modern Biology, together with Susan Holmes. And he will be discussing a couple of the topics covered in this book today. And with that, I would like to hand over to Wolfgang and get started.
Thank you very much, Julia. So great to see you all here. Welcome. So I guess why are we here? And sort of one way to articulate this in a slightly sarcastic way is this quote by R.A. Fisher, one of the pioneers of statistics. To consult a statistician after an experiment is finished is often merely to ask him to conduct a post-mortem, so you can perhaps say what the experiment died of. So a more positive way of saying the same is basically that successful, healthy, and vigorous experiments benefit from applying statistical thinking and from thinking about how you will analyze those experiments or those data right up front from the beginning. So now, many parts of this talk are in fact fairly conceptual and sort of a little bit philosophical and maybe also abstract. Normally, I actually recommend against giving abstract conceptual talks, and sort of I prefer leading by example and talking about specifics. And in fact, we do this in the book. So the current talk is essentially based on chapter 13 of the Modern Statistics for Modern Biology book, which is also linked here. Maybe somebody can also post the link to the slides that are available for this talk into the chat. So anyway, the book has 12 chapters that sort of deal with various specific statistical method areas and the applications to statistics. And then the last chapters is sort of kind of wrap up that summarize some of the ideas. And I'm now talking about those last chapters with sort of wraps up the main ideas that we have learned. And if you're looking for the more specific instantiations of those ideas, I also warmly recommend looking at the first 12 chapters of this book. So we will be talking about experimental design and sort of how this is a matter of trade-offs and good judgment. We will talk a little bit about the different types of variability, how we partition variability, about replication, sample sizes, difference between experimental studies, data transformations, also about things that we need to worry about, like dependencies, batch effects, quality assessment, and a few about computational best practices. So really experimental design is actually the art of good enough. We rationalize the trade-offs that are imposed by having finite resources. If we had infinite resources and perfect measurements, we don't really need experimental design, and everything is really easy. But of course, in reality, our instruments often have limited resolution and precision, which we don't even often know at the outset, and we have to collect preliminary data to provide such estimates for our accuracy and precisions. Sample sizes are often limited for practical, economic, and sometimes ethical reasons. And obviously the genomic data is a very good example for that, sample sizes with their samples are expensive to get and so on. If, for instance, animal experiments are involved, on the other hand, there's again ethical reasons that require us to pursue scientific questions with a minimal number of experiments that allow sound inference. Sometimes we cannot measure what we want to see directly, but we sort of have to take indirect evidence. Let's say we have a reporter of some pathway that we want, or some endpoint for a disease that we want to measure, and we need to take into account that. Often there will be nuisance factors or noise over which we have limited control, and obviously so we cannot really prescribe unrealistic ideals. We always need to make pragmatic choices that are feasible and give us good enough results. A fundamental trade-off, this is just sort of a warm-up slide, in statistics is the one between bias and accuracy, also between precision, dispersion, bias and variance, accuracy versus precision, model complexity versus overfitting. There's various different things. When you sometimes we can, for instance, increase a biomarker's false positive rate by applying a praise and false negative rate, and vice versa. We can walk through these concepts. So the obviously situation here where I'm pointing to is one where we have high accuracy and high precision. An example for high precision but low accuracy is here at the bottom left. You can see this assay, this hunter hits the right spot, hits the same spot all the time, it's just the wrong spot. Over here we have an example for high accuracy but low precision. So on average we're doing pretty well, but each individual attempt or each individual shot is a little bit all over the place. But yeah, by averaging enough, we would actually succeed. And here we have an example of low accuracy and low precision, so all over the place and in the wrong spot. But also that sort of shows you that accuracy and precision are two sort of different concepts, and they're both sort of separately important. It's also important when you plan experiments or studies to actually think about what actually you're planning. So in an experiment, everything's explicitly controlled. There's also some examples for that, and sort of the opposite, in some sense, of that in a statistical way is a retrospective observational study, where we basically take data that we get. For instance, there are study participants. We look maybe at a biobank that happens to be around in the institute, and we sort of we take the data that we could get hold of. We have often no control over study participants and or about confounding factors or the assignment of important factors of interest. So also the concept of prospective controlled studies. This is typically how clinical trials, at least sort of the big major ones, are being done, where we really prospectively assign subjects to, for instance, different arms of a trial. There's concepts of randomization and blinding. These are the most powerful and sort of the statistically, in some sense, easiest way, most straightforward trials. But it's also, I mean, they can of course be expensive, and they also can take a lot of time, because basically if they're prospective, you have to plan them and conduct them. That may sometimes mean years. So there are these constraints. And sort of a fourth major type of analysis is meta-analysis. This is so on top of everything else. We didn't even design experiment studies ourselves or collect the data. We just look at information in databases and retrospectively analyze data that happen to exist. And each of these has different statistical issues and questions, and it's good to take them hold them apart when you think about design. So in an experiment, so the sort of the typical example is a well-characterized cell line that grows in the laboratory conditions. on well-defined media, temperature and atmosphere. You administer a precise amount of a drug, and then after a specific amount of time, you measure something. So here, everything is really well controlled, and in principle, you also need very few replicates, for instance, to get the same result. If you do what you do here right, the outcome is pretty much prescribed, and there's not really much variation to expect. A lot of the other end studies, so imagine we have some, let's say, chronic disease that we want to study. We think about a cohort of patients that fulfill certain inclusion criteria, and then we, for instance, ask them to take a drug each day exactly at the same time. And before and after the study period, we perform a diagnostic test, maybe on the severity of the disease. Now, of course, what may happen, some people that we included here, of course, these are free, autonomous people. Some may forget to take the people or take it at the wrong time. Some may feel that the disease got worse in those 3 months and stop taking the drug because they don't like it. Some may feel that the disease got better and stop taking the drug because they thought that they think they don't need it anymore. There's confounding factors, so some may lead a very sort of, let's say, good, healthy lifestyle. Others smoke, drink, and eat junk food. Maybe, in fact, it's not one disease, but several. We just lump them into one bin because we don't know better. And all of these factors may even be correlated with each other in complex ways. So obviously, lots of confounding things that can go wrong. And what can we do about this? Obviously, we need much larger sample sizes. So in such a study, you may imagine we think about dozens, hundreds, maybe even thousands of participants to see reliable effects. And then there's these concepts like randomization, blinding, and good documentation of everything that happened that are needed in such a study. Sort of a recurring question that statisticians are always asked, of course, are like, how many replicates do I need? Or, I mean, everybody who plans experiments, that's of course a very fundamental question. And sort of just to bring this to a point, imagine we want to test the weight loss drugs, and I can imagine 2 experimental designs, both with 20 replicates. So we have one person that's weighed on a really precise set of scales with 20 replicates. Then he or she follows a diet, and then afterwards is weighed again on the same really high precision scale with 20 replicates, and the data are recorded by a specially trained technician. Or another experimental design, we just have 20 people weigh themselves in the bathroom scales and self-report. Which of these designs is a better one? Well, the example is absurd enough that everybody, I guess, gets the idea. And of course, the second one is more reasonable because we're not interested in the precise effect of something on a single person. We're interested in the population. And therefore, the experimental design that involves more people is more interesting, even if the individual measurements may be less precise. I mean, here we really, for instance, with the first one, we would have a very precise measurement up to the gram scale, let's say. Here, the results may be even off by some kilograms. But in the end, that experimental design would still give us, in the end, the more useful data. So again, this may sound silly, but actually there are quite many design decisions in high throughput biology that are similar, that are maybe a bit more subtle, but in the end boil down to the same kind of situation. So when you want to, for instance, when your replicates involve, for instance, producing several sequencing libraries of the same sample, do you want to use one cell line multiple times in replicates, or do you maybe want to use several different cell lines, each with only one replicate? If you have a certain target genes you want to target, do you just use one CRISPR guide or lots of different CRISPR guides for the same gene, and how many? Or if you want to, for instance, put up a certain pathway with a drug, do you use one drug and replicate a lot, or do you use several drugs, different drugs, and experiment that way? And there are obvious trade-offs here that sort of come down to the same type as in sort of this very idealized or stylized example in the beginning. So to make this more concrete, the the human, we all know that for reliable variant calling, let's say with the sort of the most common current sequencing technologies, we need about 30x coverage of sort of short reads to cover a normal human deployed genome and to get reliable variant calls. In the 1,000 Genomes project, which is a big landmark study that was performed, I think now some 15 years ago, the average depth, so that they did also sequence 1,000 human genomes at the time, but they only sequenced each to about 5-fold coverage. So why did they use the study design? If, for instance, if we had the 30x coverage, here obviously was very much underachieved. Why was that study design chosen anyway? I guess the answer is obvious. We're not in the end interested in a single person's variants. In the study, they were interested in population level variants that by definition occur in multiple different individuals. So let's say if your definition of a SNP that has a frequency of 1% or more in the population, they would expect about 10 people to have a certain SNP at this 1% frequency, and therefore they would still get a coverage of about 50x for any SNP of interest. And therefore that design was chosen. So the opposite, like sort of the other sort of obvious choice would have been, for instance, to take 170 individuals and do each of them 30x coverage at about the same cost. So here you see those kinds of thoughts or trade-offs. How do you allocate precision per subject, per sort of replicative unit, can have important consequences. So what exactly is a technical versus a biological replicate? And in fact, the terminology is really not that useful, and we should probably drop it, because variation can occur at many different levels. So for instance, as I mentioned, you can have different CRISPR guides for the same target gene. We could have different drugs for the same target protein. We could have different cell lines or different organized animal models for the same biological system, let's say for the same disease. We could have used different measurement technologies, because for instance, we could look at the same tumors using multiplex immunohistochemistry or imaging mass cytometry, and so on. We could use different machines, for instance, different sequencing machines for the same technology, or different variants of the same protocol, and so on, different technicians in labs. So as you may imagine, at all of these different levels, there's a potential for application. There's room to think about which of these factors we vary and to what extent, and where we stop varying, because at some point, of course, the experiment has to stay finite. But those are far more subtle and detailed than just the difference between a biological and technical replicate, and therefore it is useful to think about this in this much more detailed way. And in any case, we then use the ideas of blocking and the tools of design matrices and linear models to account for this. I will come a little bit back to this in the following slide, what I mean by linear models and design matrices. So to the extent that we know about all sort of extra factors, like which cell line, which measurement instrument, which technician, we of course need to keep track of such factors and include them in the metadata of our data, and also maybe then explicitly as bias terms in our analysis models. Sometimes we did not keep track of all of such different factors, and we may still, in high throughput data, actually we often have the luxury, or in a good situation, that we may be able to use latent factor models such as SVA or PIA or RUV, that we can identify such what we call batch effects back from the data, because we have so many different features. For instance, in RNA-seq or in imaging, we have a lot of very high dimensional data, and may be able to estimate back some of these factors, even if we lost track of them. Which brings us to the topic of error models and linear models. I already mentioned this. Here, just sort of to start that, the efficiency of most biochemical or physical processes that involve nucleic acids, so for instance, DNA sequencing or RNA sequencing, depends on their sequence content. For instance, the occurrence of long homopolymer stretches of also GC content. I mean, it's a pretty sort of common law of biochemistry, bioinformatics, that everything depends on GC content, whatever you look at. But those effects also are not universal. They may also depend on concentrations, temperature. So that the effect, for instance, the effect of GC content on the sequenceability of an mRNA or cDNA is not a universal constant. It can depend a lot on details of the protocol that was used for the RNA-seq experiment, and therefore that effect may actually be different from experiment to experiment. So, should we then try to model this when looking at RNA-seq? That should we treat GC content just as a pure noise term that we ignore, or should we sort of include it as a covariate for each gene and sort of try to model that as a factor? And of course, there is no right answer. Both are possible, and there are, for instance, bioinformatic methods for looking at RNA-seq data that ignore GC content and basically hide it in the error terms. There's other models that take it into account explicitly and sort of try to estimate parameters for GC content. And both are valid things to do. And sort of a very placative example of that is one person's noise can be another bias is an experiment that was done by Percy Diaconis. So here he's showing basically a coin tosser. Coin tossing is obviously the prototypic example of something that is completely random, supposedly completely random. But in fact, if you can basically, if you exactly register the speed and the rotational momentum and all of the different physical factors and the air drag of a coin when you throw it in the air, you may in fact predict how it lands. And they even did experiments to prove that. So depending on how much attention you pay, you may either treat coin tossing as something completely random, where both outcomes have probability of a half, or if you, for instance, record all of these different initial conditions and do the physics, you may actually be able to use modeling to predict those. And which one of you do is really your choice as an analyst. So noise models, error models are really not an objective fact of the world. They're sort of a subjective measure of our ignorance, either willful or intentional. One thing that really, compared to physics, of coin tossing, that really confounds biology is the lack of units. And I like to motivate this sort of by looking at pre-modern measurement systems, where we, for instance, measured lengths in feet, ells, or inches, which is actually the word for the first joint of an index finger. Weights are measured in stones, volumes are multiple of the size of a wine jar, and so on. As you might imagine, those units were very useful and sort of reproducible based on certain prototypes or artifacts, but it could very well be that let's say an English inch would be different from a Franconian one, and again different from a Swiss one. So there was all of these sort of batch effects in measurements, and you couldn't really compare measurements across different places, for instance. On the other hand side, there was some value to these measurement systems, and then they were internally perhaps coherent. In the International System of Units, which is a development sort of that happened in the 19th century, the units like meters, seconds, and so on are defined based on universal physical constants. And in fact, one of the wonders of physics is that there's no reliance on artifacts here. And a meter can be measured by a lab in Canada using an instrument and has the same meter as a meter measured way later by a lab, let's say in Heidelberg, using a very different instrument, or in fact by a space probe out somewhere in the solar system, or even by an extraterrestrial intelligence on a completely different planet. As long as we all agree on the physical definition of a meter, those measurements would be comparable, and even without exchange of artifacts like a meter prototype. So that's a really beautiful situation that physics is in. In biology, the measurements are unfortunately rarely that kind of comparable. Most biological assays provide measurements in some arbitrary units, fluorescences or read counts or whatnot. And therefore, we don't really have such references. So this is true even if we have absolute values like read counts, then they're unitless, because they still don't translate into universal units such as like molecules per cell or moles per millimeter and so on. So that's one of the problems here. The solution, the perceived solution, is called normalization. This is really everything that we need to do to deal with the lack of units in many biological assays. So we try to make measurements comparable to each other, at least internally within one experiment, using some arbitrary but at least consistent units. So for instance, we try to bring the read counts from our different cell in our single cell experiment to the same scale by sort of rescaling them, so that at least the value of 1,000, let's say, is comparable across different cells, even though within the experiment, even though it doesn't still mean anything universally, and it would be not comparable to a value of 1,000 measured by a different experiment. But so there's this sort of trying to make things internally consistent. Another sort of way to address this is to only consider endpoints where the units cancel out, for instance, by looking at fold changes, rather than the absolute values. And they also can work to some extent. So examples already, so obviously everything that becomes measured in sequence counts, for instance, because it will depend on library size and sampling efficiency of the instrument. Those may vary a lot. So there's no easy relationship, let's say, between moles per cell and the count table. Same for mass spec intensities, same for fluorescence intensities, let's say in an immunohistochemistry assay. The measurements are sort of internally comparable to some extent, but not really universal. By the way, normalization is really a terrible name for this concept. So I defined what it is in this previous slide and in the slide here. And in some sense, probably the word calibration would be a better name, or I don't know what a good name would be. Normalization it has nothing to do with normal distributions or vector norms or right angles. But still, it's the name that has stuck and is sort of being used a lot in the field. So this is just an example from RNA-seq. What you see here is an RNA-seq experiment. On each point corresponds to a gene. Actually, this is plotted using hexagonal binning. So basically, the density of points is indicated by the color code as well. There's two different samples being compared. On the X and the Y axis, and sort of you see the differential expression analysis between those two different samples. If I just, and this is on a kind of log-like scale of the counts, and if I just, for instance, take the data at face value, and also draw in the intersect line, I see that, for instance, the counts for the 517 sample are consistently a bit higher than the counts for the 513 sample. You see the layer lie a little bit above. This could be biologically real, or it could just be that the counts in general just all a bit higher. Maybe they're all higher by a factor of 1.5 because we simply sequence deeper for this library. So it's very tempting then, for instance, to rescale the values for this second sample to bring the values on a comparable scale, and then, for instance, to make most numbers, most counts agreeable or come close to the orange line, and then just look at at those values where there's really a big distance from the orange line. Yeah. So as you can see, this is a step that tries to minimize, tries to account for the lack of units in those data. If we don't do the normalization, or if we do it wrongly, we can perceive increased variability between replicates. So for instance, we could come to the conclusion that all the genes or many genes in the second sample are upregulated. And this can, of course, produce false conclusions. What we want from a good normalization method, that it removes such technical variability, that it keeps biological variation. But it is really hard because there's there's often no easy way to decide between multiple different normalization options. This requires some sort of often some back and forth. We can compute figures of merit, we can measure, for instance, how good the normalization actually works. We can look at things like noise ratios, for instance. We can look at positive and negative controls in our experiments, and then, for instance, expect that between negative controls and between replicates, there should be no differences left after normalization, whereas the difference between positive and negative controls, for instance, should still be large, and so on. A sort of underlying thought here is sort of maybe also encoded in Occam's razor. If I can explain a phenomenon without assuming a hypothetical entity, that's no good for assuming it. For instance, if I here in this case said that, wow, this data indicate that all of the genes in the second sample are upregulated, something really amazing is going on here. Also, the easier explanation is that I just sequenced that second library a little bit deeper, but that in fact there's not much going on between these two different samples. Then, of course, the second explanation is somehow is more coherent, more consistent with Occam's razor. That's why it's a sort of a useful concept. So if in effect can be normalized away using a sort of reasonable idea of the measurement process, then it's not interested. We should mostly focus on things that cannot be normalized away. In the context of noise modeling and normalization, it's also important to distinguish between regular and catastrophic noise. So regular noise we can often model by a simple probability model, like let's say the normal distribution or the Poisson and so on. We can also regular normalization sort of factors. We can, for instance, as I mentioned before, for the RNA-seq, we could, for instance, adjust for sequencing depth using by dividing all of the counts by a certain factor, and so on. So we can basically use relatively straightforward methods to take such regular noise and regular biases into account. But the real world is also more complicated. Sometimes we have catastrophic events. Measurements can be completely off the scale. There maybe have been a sample swap. Maybe the sample was contaminated. There was a software bug, and all the values have been sort of inverted or something. And in those cases, in fact, no error model, no simple error model and no normalization will help. And it's then really important towards that error models and normalization methods realize their boundaries and reject then dealing with such outlier or catastrophically failed data. Another thing is that multiple errors often come together. So for instance, if something goes wrong, often go, and this is we know this very well from everyday life. Something goes wrong, it often goes wrong together in concert. So for instance, errors don't always occur sporadically. For instance, the whole microdata plate can go bad at the same time. And how do we keep track of this? So I just want to sort of highlight this example that is from a collaboration on an RNA-AI screen that we performed some time ago. So what you see here are basically data from, I think, 60 384-well plates. Each of the little boxes here corresponds to one 384-well plates, and the wells, there's 384-well pixels in each of those little gray boxes that corresponds to the 384 wells in the plate. So altogether, this is a genome-wide screen. If you multiply the number 60 times 384, and each well corresponds to different, in this case, RNAi treatment for a gene. And I think what we measured here was basically the response of a certain cell line to those to those many different tens of thousands different of RNAi treatments. So far, so good. But if you now plot and the data, when you get the data, of course, it's just one big Excel table of sort of siRNA IDs and viability measurements. If you plot the data in this way, by plate, or by physical location, you see a lot of different things. You see what I mentioned before. Sometimes whole plates go wrong. Somehow the measurement errors does not look uniform. It does not. Some of the measurements seem to be very highly correlated. For instance, at the bottom here, there's 3 plates where everything is really dark, meaning really high values. For those 3 plates, we see edge effects that sometimes values at the edges of the plates are high and in the middle are lower. We see row effects that, for instance, in this case, the first 2 rows somehow have strangely high values. Then all the other values are more or less normal. We see checkerboard effects that maybe come from the fact that the 384 well plates were all combined by piping together 496 well plates in sort of an interspersed grid. So we see all sorts of experimental artifacts and patterns here that are indicative of correlated and in some way catastrophic noise. In the end, this data had to be thrown away because the artifacts were just too many, and it wasn't possible to find an error model or a normalization method that could really deal with these data and remove the artifactual normalization without removing too much of the biologically interesting signal. Unfortunate thing here is that this experiment, I think, took some postdoc, several weeks. Like, think about each plate, I don't know, 2 or 3 days of incubation time, and it's done one after the other. And they just did the whole experiment basically in one long go, and then looked at the data in this way. I mean, this was the first, only a post-hoc. We got the data table and plotted it in this way. And then, of course, it was too late to do very much about the data. This comes back to the idea of keeping track and basically doing dailies. So this is something where we know from actually from Hollywood, from film taking, that basically a film director will also not constantly take shoots and have films taken with footage taken without constantly sort of looking at the material, and for instance look at any potential lighting or shooting issues. So that's the idea of dailies in Hollywood, and we should have this. We do have the same concept in sort of high throughput experimentation, that we should basically monitor experiments as we do them. And have a constant and continuous data production surveillance while data are being produced, so that we don't have the problem that I mentioned before of just discovering such problems post hoc when it's too late to fix them. Sounds easy and obvious, but of course in any sort of really large experimental campaign, this can be hard, and it's quite a bit of extra effort to actually implement, as for instance, also Julia can attest to. I will go on for about 5 more minutes or so, I think. One sort of fundamental principle of experimental design that is sort of very useful, actually goes back almost 1,000 years now, back to Avicenna or Ibn Sina, that when you want to look at experimental data or study data, that well, you need, of course, control some replication, that you should also only vary one factor at a time, that this is sort of gives you the most robust inference, rather than varying lots of different things concurrently. In fact, this dogma has a little bit overdrawn in the 20th century by R.A. Fisher, where with the theory of linear models and design matrices, where there's in fact an analysis of variance, but in fact it is possible to look at experiments in meaningful ways where we have covariation of multiple factors at the same time. So let's, for instance, look at this experiment that I'm sort of showing here. We have some measurement in biomarker Y, measured on 6 different individuals under 2 different conditions. As you can see here, there seems to be quite a nice difference between the 2 conditions. If you do, let's start here. here we have such a comparison. As you can see, there's a nice difference between those two conditions. And if you do any statistical test, this would be highly significant. But now, however, suppose the experiment was done in 2 batches. And if we, so for instance, using 2 different sets of measurement instruments or sets of reagents, and it may be the fact that those 2 batches are completely confounded with the condition, then we actually would not be able to conclude anything, because the confounding factor, something that we should normalize for, is completely masking our experimental effect. So that's one example of a bad experimental design. But let's assume that we actually have taken care of this, and that that any unknown batch effects are balanced between the conditions. What then happens is that those unknown batch effects still can cause higher noise levels. For instance, because our measurement instruments always measure more higher values on Fridays than on Mondays. But if we did good balancing of all of these factors, this would only would affect both groups simultaneously, but it would in effect lead to an increased noise level. And now, for instance, in what you see here, the noise level would have been increased so much that the comparison, even though it has the same effect size, is no longer significant because of the higher noise. In that case, we would actually need to take a larger sample size. So if we then do the same measurements again, but using 24 replicates, in fact, would again be able to detect the difference using the higher sample size. So the lessons from this toy example is that the success of an experiment depends not only on effect size, but also on the underlying noise factors. And one way of increasing our detection power, our success, is in fact if we are able to decompose such variability. So one way of dealing with the variability is to treat it as noise, and that's what is shown here, and what gave us the bad p-value. If we know those batch effects, so for instance, if we knew that we had measurements were done in 3 different batches, 0, 1, 2, that are encoded by these colors, we may actually be able to model those batch effects. And if we do that, and this is shown here now with these 2 different linear models, with the same data, if we just compare the 2 different conditions, so this is now a little bit of R code, but this basically what it means is we do a test between those 2 different conditions, we see the noise is so big that the effect size, that the condition effect is masked or not doesn't raise above statistical noise. If we are actually able to model this batch effect and include it in our model, so now I'm formulating a 2-way ANOVA on the same data with this additional batch effect, then suddenly we see that this condition effect becomes, it gets a small p-value, so it becomes significant. And that's an instance, an example of how modeling of noise, of how taking something that was noise in one condition, but modeling it and taking it explicitly into account can improve our analysis. And I will just now do the paired experiment, and then maybe conclude and go to the questions that have come. So a special case of such taking into account such blocking factors or batch effects, if you wish, are paired experiments. So this is an example. This is the famous Darwin sea mice experiment, where he measured the effect of different pollination methods, or 2 different pollination methods, on plants. And in this case, because of course, obviously the plants also grow depending on many different factors, like how much light they get, how much water they get. So if you just look at individual, basically there's lots of additional confounding factors that are potentially existent. So what you did here is he actually took 15 different pairs of plants, each with 2 different pollination methods in 4 different pots. And in this case, it's paired design, where basically each pair of plants could be compared for cross and self-pollination. It's a special case of such an ANOVA type analysis. What you can do here, if you have such paired measurements, you could do a paired t-test. And in fact, it turns out to be equivalent to a two-way ANOVA where you have type and pair. And this is just here exemplified by this example R code. You can see that the results that you get, the estimated effect sizes, and also the p-values are exactly equivalent between the two-way ANOVA using the linear model and the paired t-test. Sort of last comments on the t-test. There's often a lot of anxiety about using t-tests in data analysis. There's a proof that the t-statistic follows a certain distribution, which is called a t-distribution, which assumes that the observations are independent and follow normal distribution. And it's important to keep in mind, and people often get very queasy about those assumptions and sort of think whenever the assumptions are not being met, test is invalid. That's not actually true. The assumptions are sort of made, they are sufficient to make the math simple and tractable. The sort of sufficient conditions, but they're not necessary in a logical way. They're actually sufficient. So the test may actually be valid under a much wider set of scenarios than just those that are used to prove the mathematical fact. And in fact, deviation from normality, so heavier tails, and very few cases a real problem. The test may lose some power, but the test still remains valid in terms of its type 1 error control. Whereas deviations from independence of the data, which is the second part of the assumption, this is really critical. And for instance, if the data are not independent, there's some correlation between measurements, then in fact the p-values or any conclusion from a t-test can become totally wrong. And what I often get in sort of collaborations or talking to experimentalists is actually the complete opposite. People are really worried about normality and do all sorts of things to sort of worry about this, and are completely unworried about independence. So batch effects, confounding factors, and in fact, it should be the opposite. So like relax about normality assumptions and be really apprehensive about any correlations in the data. I have a little app that shows this. So what I'm showing you here is the null distribution of a t-test or of on null data. Well, first using on normal data. And as you can see, it's uniform as it should be. These are the p-values. If I introduce correlation between the variables, just correlation, so still no effect size. On average, there's nothing happening in these data. And the data actually shown on the right-hand side, so you can sort of look at this at your own pace afterwards. You can see I get an enrichment of small p-values. Even the slightest correlation between data leads to sort of an over-optimistic test and in this case, invalid p-values. Whereas if I change the distribution of the data, log normal, exponential, uniform, whatever, the histogram hardly changes. There's a little bit of differences, but in any case, I don't get this high enrichment of small p-values. Only correlations give me create a real problem. And with this, I see it about 15 minutes, and maybe it's time to respond to some of the questions in the talk. I'd like to acknowledge for the input to this talk Susan Holmes, who's also the co-author of the book, and also Julia Philipp, who made a lot of useful inputs to the slides.